Welcome back to the deep dive. We've got a fascinating topic today, digging into, well, one of the most elegant biological logic systems out there.
That's right, we're tackling bacterial gene control, specifically the operon.
Right, how these tiny single-celled organisms manage this huge amount of genetic information so efficiently.
Exactly. Think about it. They need to decide right now which genes to turn on, which to turn off. Our mission today is unpacking how they control this at the very start, at transcription initiation, focusing on these bacterial operon.
Okay, so let's just quickly recap the starting point, the central dogma, right? Transcription needs RNA polymerase to find the promoter sequence on the DNA.
Precisely. And in prokaryotes, it's pretty streamlined. You have the RNA polymerase, but it needs help. It relies on this crucial helper called the sigma factor.
Sigma factor, right?
Yes, sigma recognizes specific sequences in that promoter, the -35 and the -10 regions. That -10 site, it's rich in A's and T's, so it's easier to unwind.
Ah, okay.
So sigma basically acts like a GPS, positioning the polymerase perfectly to get started.
So the promoter says transcription can start here, but something else decides if it will start. That's the regulation part.
That's the core logic, yes.
Right.
The cell isn't just randomly transcribing everything. It categorizes its genes based on need. Three main types.
Okay.
First, you've got the essentials, constitutively expressed genes. These are always on. Think tRNA genes, ribosomal RNA, the proteins that make up ribosomes, RNA polymerase itself, kind of the basic housekeeping
The
stuff.
non-negotiables, always running.
Exactly. Then you have the genes that are only needed sometimes.
The conditional ones.
Right. Two types here. First, inducible genes. These are normally off, silent. They only get turned on when, say, a specific molecule appears in the environment.
Like a new food source.
Usually, yeah. Something the cell needs to break down. So they have to be induced, switched on.
Okay. And the third category?
Repressible genes. These are the opposite. Normally on in, they're busy making essential building blocks, maybe amino acids.
Stuff the cell needs to build itself.
Right. But if that building block suddenly becomes available from the outside, or if the cell makes too much,
It needs to shut production down.
exactly, it represses those genes, turns them off, saves energy.
So turning on the inducible ones, turning off the repressible ones. This control relies on specific proteins, right? Transcription factors.
That's the toolkit. These are DNA-binding proteins. They have specific domains, maybe 60, 90 amino acids long,
Yeah.
that recognize very particular DNA sequences. Non-covalent binding, usually.
And they act like switches.
Pretty much.
Yeah.
They come in two main flavors. Activators, which help recruit RNA polymerase, turning transcription up or on,
Okay.
and repressors, which do the opposite. They block or inhibit the polymerase. Gatekeepers.
This is where bacteria get really cluttered with the operon structure. Tell us about that.
Ah, the operon. It's brilliant for efficiency. Instead of having separate switches for genes needed for the same pathway, bacteria often cluster them together. Genes A, B, C, maybe, all controlled by one single promoter.
One promoter, okay.
And crucially, one regulatory site called the operator. The whole cluster gets transcribed as a single messenger RNA molecule.
One switch controls the whole production line. That operator. Where does it sit?
Strategically placed. It usually overlaps the end of the promoter at the beginning of the first gene, right where transcription starts.
So if something binds there,
It can physically block RNA polymerase from moving forward. That's the basis of negative control, where the regulatory protein is a repressor. It acts like a stop sign.
okay, negative control uses a repressor, and you mentioned two types, inducible and repressible.
Yes. Let's break those down. First, negative inducible operons. Think of this as needing to take the brake off.
Okay, so the default is break on.
Exactly. Transcription is normally O off F because an active repressor protein is already sitting on the operator blocking things. This setup is common for pathways that break down substances from the environment.
So how do you get it started?
A specific small molecule, called an inducer, needs to show up, usually related to the substance the pathway breaks down.
The key to unlock the brake.
Right. The inducer binds to that active repressor protein. This causes the repressor to change shape. It basically makes it let go of the DNA.
Ah, conformational change.
Yes. The repressor releases the operator. The polymerase can now bind and transcribe the genes. Break off, pathway on.
Makes sense. Now the other side, negative repressible operons. This is slamming the brake on.
Correct. Here, the default state is on N. Transcription is happening because the repressor protein, in its basic form, is inactive. It can't bind the operator on its own.
So the pathway, maybe for building something essential, is running.
Right, like synthesizing an amino acid. But what if the cell gets enough of that amino acid?
Needs to stop making more.
Exactly. The end product of the pathway, say the amino acid tryptophan, acts as a signal. It functions as a core pressor.
Core pressor, meaning it helps the repressor.
Precisely. The core pressor molecule, tryptophan in this case, binds to the initially inactive repressor protein.
And that binding activates it.
Yes. It forms an active repressor-core pressor complex. This complex can now bind tightly to the operator sequence.
Blocking transcription, slamming the brakes on.
Exactly. The classic example is the trap operon in E. coli, which makes tryptophan. When tryptophan levels get high inside the cell, tryptophan itself acts as the core pressor, activates the trap repressor protein, and shuts down its own synthesis pathway.
Wow. A perfect feedback loop. Prevents wasting energy.
It's incredibly efficient. The cell only makes what it needs when it needs it.
Okay, let's dive into the most famous example, probably. The Lac Operon. This is the poster child for negative inducible control, right? How E. coli deals with milk sugar, lactose.
That's the one. The Lac Operon has the genes needed to metabolize lactose. Key players are LAC-Z, which makes an enzyme called beta-galactosidase.
That breaks lactose down.
Yes, into glucose and galactose. It also does a little side reaction, converting some lactose into allolactose. And then there's LAC, which makes permese, a transport protein to get lactose into the cell. There's LAC-A too, but its exact role is a bit less clear.
So in the standard state, no lactose around.
The LAC-I gene, which is separate from the operon, constantly produces an active LAC repressor protein. This repressor binds very tightly to the operator site, LAC-O.
Blocking transcription of LAC, Z, Y, and A.
Mostly, yes. It significantly inhibits RNA polymerase. But here's a really crucial point. It's not a 100% perfect seal. There's a tiny bit of leakiness.
Leakiness. Why would the cell allow that?
That's the brilliant part. Think about it. If repression was absolute, zero permese, LAC would be made. If there's no permese, how could lactose ever get into the cell to signal its presence in the first place?
Right, you need a sensor. So the leakiness ensures there are always a few molecules of permese in the membrane.
Exactly. And a few molecules of beta-galactosidase, LAC-OZ, inside, it's just enough. So when lactose does appear outside
A little bit gets transported in by that leaky permese.
Right. And inside, the few beta-galactosidase molecules convert some of that incoming lactose into allolactose.
Now, allolactose is the actual inducer molecule, not lactose itself.
Correct. Allolactose is the true inducer. It binds to the LAC repressor protein that's sitting on the operator.
Causing it to change shape and let go.
Precisely. The repressor pops off the DNA. Suddenly, the path is clear for RNA polymerase. It binds the promoter, and boom, transcription of the LAC genes skyrockets.
How much of an increase are we talking?
It's dramatic. Within minutes, you can see something like a 1,000-fold increase in the production of these enzymes. It's called coordinate induction. All the genes turn on together, massively.
Okay, so the cell can turn on lactose digestion, but what if it has a choice? What if there's lactose and glucose available?
Now we get to the next layer of control. Cells are smart, metabolically speaking. Glucose is the preferred energy source. It's easier and more energy efficient to metabolize directly.
So even if lactose is present, if glucose is also there, the cell would rather use the glucose.
Absolutely. It prioritizes. This phenomenon is called catabolite repression. Basically, the presence of the preferred catabolite glucose represses the operons from metabolizing other, less preferred sugars like lactose.
So how does it enforce this priority? Is this another negative control thing?
Actually, no. This layer is a positive control mechanism. It enhances transcription of Lac Operon, but only when needed. It works alongside, or maybe on top of, the negative control system we just discussed.
Positive control. So it boosts transcription.
Exactly. It asks the question, is glucose scarce? If the answer is yes, it provides a boost to the Lac Operon, assuming lactose is also present.
Okay, how does this positive boost work?
It involves two key players, a molecule called CAMP, cyclic AMP, and a protein called KMP, catabolite activator protein.
CAMP and CIP.
And here's the crucial link. The level of CAMP inside the cell is inversely proportional to the level of glucose.
Inversely proportional. So high glucose means low CAMP.
Correct.
And low glucose means high CAMP.
Exactly. When the cell is low on glucose, its preferred food, CMP levels rise significantly.
Okay, so high CAMP is a starvation signal for glucose.
Right. And when CAMP levels are high, CAMP binds to the CIP protein. This forms an active CIP complex.
And this complex is the booster.
Yes. This CAMP2 complex binds to a specific DNA site located just upstream of the Lac promoter.
What does binding there do?
Its presence physically helps RNA polymerase bind much more effectively to the promoter. It sort of bends the DNA a bit, making the promoter more accessible. It acts like a supercharger, increasing the rate of transcription initiation by up to 50-fold.
Wow, okay, so let's put it together. For maximum Lac Operon expression.
You need two conditions met. One, lactose must be present. So allolactose forms and removes the Lac repressor that's releasing the negative control brake.
Condition 1, Burkhoff.
And two, glucose must be absent. So CMP levels are high, the CMP2 context forms and binds, providing the positive boost.
Condition 2, booster on.
Exactly. If glucose is high, CMP is low. No KMP context forms. So even if lactose is present and the repressor is off, transcription from the Lac promoter is quite inefficient, quite low. The booster is off.
That's really elegant, a two-factor authentication system for turning on the lactose genes fully.
It ensures the cell focuses its resources on the best available fuel.
And a lot of this intricate detail was figured out by studying mutants, right, when things go wrong. Jacob and Monod's work.
Absolutely foundational work. By studying E. coli with mutations in different parts of the Lac system, they pieced together how it all worked, identifying the structural genes, the promoter, the operator, and the repressor gene.
And it's led to understanding different types of regulatory elements.
Yes. They made the crucial distinction between cis-acting elements and trans-acting factors.
Cis and okay, what's the difference?
Cis-acting elements are DNA sequences that only control the genes located on the same piece of DNA. They have to be physically connected. The promoter, LAC-P, and the operator, LAC-O, are classic examples. They don't make a product that floats away. Their function is tied to their location.
They affect the genes right next to them.
Right. Whereas trans-acting factors are typically proteins. They are produced from genes that can be located anywhere else in the genome. The protein product then diffuses through the cell and can act on any DNA molecule that has the correct target sequence.
Like the Lac repressor protein itself.
Exactly. The LAC-I gene produces the Lac repressor protein. This protein can then float over and bind to the LAC-I operator site. Even if LAC-I is on a completely different piece of DNA, like a plasmid, it acts in trans.
The cis-trans distinction helps make sense of the mutations they found. What happens if you just break the repressor gene, LAC-I?
A simple loss-of-function mutation called LAC-I. The cell can't make a functional repressor protein.
So, no break.
No break. The operon is always on-in or constitutive. It transcribes the Lac genes whether lactose is there or not.
And what if the operator site itself is mutated, LAC-O?
Same result. LAC-O means constitutive operator. The operator DNA sequence is changed, so the repressor protein, even if it's perfectly normal, simply can't bind to it anymore.
Can't grab hold. So again, always on in.
Right. Both LAC-I and LAC-O lead to constitutive expression. But studying them in cells with multiple copies of the Lac region revealed the cis-trans difference.
Ah, okay. But the really telling mutation is the super repressor, isn't it?
Oh yeah, the LAC-I mutation. This one's fascinating and dominant.
Dominant, meaning it overrides the normal function. How does it work?
The LAC-I mutation creates a repressor protein that has a subtle change. It can still bind perfectly well to the operator DNA.
Okay.
But it can no longer bind the inducer, allolactose.
Oh. So even if lactose is present and allolactose is made inside the cell?
The superrepressor doesn't care. It ignores the signal.
Yeah.
It stays firmly clamped onto the operator sequence, blocking transcription permanently.
Regardless of the environment.
Exactly. The result, the cell cannot metabolize lactose, ever. The operon is permanently O off F. Because this faulty repressor protein is transacting, even one copy of the LAC-I gene in the cell is enough to shut down all functional Lac operons. That's why it's dominant.
Wow. That really demonstrates the power and mobility of that repressor protein.
It does. It shows how crucial that interaction between the repressor and the inducer is for the whole switch mechanism.
So wrapping this up, this deep dive really shows an incredible multi-layered system in bacteria. It's not just an on-off switch.
Not at all. You've got the negative control of the repressor responding to the presence or absence of the substrate, like lactose via allolactose. That's the primary switch.
And then layered on top, you have this positive control, the catabolite repression system with CA-key and CAMP. That's like a dimmer switch or a priority check.
Right. It fine-tunes the expression based on the cell's overall energy status, prioritizing glucose. It's all about efficiency, minimizing waste, and reacting quickly to environmental changes.
The elegance really is in that efficiency. Nothing wasted.
Precisely. And think about that super repressor again, the Lac Operon.
Yeah.
We saw how one small failure in molecular recognition, the repressor, losing its ability to bind the inducer, leads to a dominant, catastrophic failure for the cell in using lactose.
Yeah.
It can't adapt. So here's something to ponder. What does the very existence of these kinds of highly dominant failure states tell you about the intense evolutionary pressure there must be to maintain absolute precision, that perfect lock-and-key fit in these regulator proteins and their binding sites? Something to think about, reflecting on the molecular logic running inside every cell.